Thank you very much. It is really exciting to be here. So thanks to the organizers for the opportunity to present my PhD work looking into the mechanisms regulating epithelial cell plate plasticity. And this is unpublished work. So epithelial tissues cover the surface of our body with the skin and also the internal cavities of our organs, such as that of the esophagus, the airways, or the intestine. Their main function is to act as a barrier that protects us from external aggressions. And therefore, it makes sense that epithelial cells have adapted to respond very rapidly to tissue perturbations. And one of those mechanisms is cell plasticity, that allows lineage-committed cells to reshape their cell fate in response to tissue damage. In response to tissue injury, we know that the tissue microenvironment, or its niche, is critical at orchestrating cell plasticity events. For example, this pioneering study from the 70s used a chick embryo to show that areas of the epithelium that don't have feathers can actually start developing them by simply exposing them to the stroma of skin that has feathers. This work was followed, well, these kind of studies were followed up over the years, and more recently, the Fuchs lab showed during mouse embryo development that the epithelium is competent to form either hair follicles or sweat glands by simply regulating stromal signals. So these examples, this phenomenon of identity switch, have been particularly described in developing tissues, which are by definition more permissive, but have barely been studied in an adult context. So with this project, we wanted to investigate what are the molecular mechanisms and the temporal resolution of adult lineage conversion, which obviously has potential implications for regenerative medicine. So to address these questions, we chose our two favorite tissues, the esophagus and the tail skin, which are very similar from an architectural point of view, but have the key difference that while the esophagus doesn't have any appendages, the tail skin has different stem cell compartments like hair follicles and sebaceous glands. So we took advantage of that difference to ask whether esophageal cells could respond to the skin microenvironment, basically whether we could switch tissues and convert esophageal epithelial cells into skin cells. For that, we adapted a model that we had used in the past to study wounding in vitro. And the advantage of this model is that we can combine epithelium and stroma from different origins and different animals, allowing us to study the cell behavior in a newly formed epithelium. So in this case, we grew two strips of esophageal epithelium on top of the skin stroma of the dermis of another animal. To identify cells coming from either tissue, we harvested the esophagi from animals expressing red fluorescent protein and the dermis from DFP reporter animals. And the resulting 3D heterotypic culture is then kept in vitro under minimal media conditions. So this is without any added growth factors. And the newly formed epithelium can then be compared to that of our controls, where the esophageal strips are grown on top of their native stroma. Following 10 days in culture, we were surprised to find that esophageal cells in red hadn't only covered the dermis, but had also colonized the underlying hair follicle sockets and sebaceous glands, forming these structures that I will refer to as esophageal-derived hair follicles. And this wasn't an isolated event, but actually over 90% of the hair follicle sockets had been re-epithelialized by esophageal cells. So next we wanted to investigate the cell identity within those structures, and for that we looked into hair follicle markers. For that, we used a set of controls, including the tissue of origin, the esophagus, and the host tissue, the skin. As you can see in the top panel, none of these hair follicle markers was expressed in the esophagus in vivo, and this was also the case for the esophagus in vitro. However, when we looked into our esophageal-derived hair follicles, we found that over 75% of them expressed keratin 17 and keratin 24, which was very promising. However, when we looked into a well-defined marker of hair follicle stem cells, CD34, we were shocked to see that only about 25% of them expressed this marker, hinting us to the existence of potential barriers to lineage conversion. So to explore what was either favoring or preventing lineage conversion, we turned to single-cell RNA sequencing of cells grown on the stroma of either tissue, the esophagus or the skin. And to have a temporal overview of the process, we looked at two different time points. We found that at the early time point, both cell types shared a very similar transcriptional signature that was characterized by a strong regenerative profile. However, at the final time point, we found two different profiles. As anticipated, esophageal cells grown on their native stroma had shifted towards the bottom of this UMA projection and showed a more differentiated state, while cells grown on the skin stroma remained locked in an intermediate area, and they partially retained the regenerative signature that we had found in the earlier time point. To explore in more detail this apparent blockage, we performed a pseudo-time analysis inferring the trajectory of these cells from the esophagus to the skin identity. And as you can see, cells grown on the esophageal stroma and skin cells separated at both ends of this trajectory, while cells grown on the skin stroma formed a smear of cells moving along this trajectory. So we analyzed these three different groups, the two with a major esophageal or skin identity, and the one with a significant proportion of cells in this transition. And we analyzed the genes that were significantly differentially expressed in this region, in the transitioning cells. And we found something that really stood out was the hypoxic signature of these transitioning cells that was defined by an upregulation of HIF1 alpha inducible genes. So then the question was, is HIF1 alpha promoting the cell conversion or is it preventing it? To address that question, we used HIF1 alpha modulators and analyzed the expression of CD34 as a readout of lineage conversion. We found that while HIF1 alpha activator was abolishing CD34 expression, its inhibition was actually boosting it, suggesting that HIF1 alpha is acting as a barrier to lineage conversion. We were also interested to look at SOX9, which is a well-defined transcription factor involved in plasticity, and that has been described to act downstream of HIF1 alpha. And we found that indeed in our model, also HIF1 alpha and SOX9 go hand in hand, suggesting that SOX9 may be regulated by HIF1 alpha. We then asked whether SOX9 by itself could have an effect on the lineage conversion phenotype. And for that, we used an animal model where we can conditionally delete SOX9 in epithelial cells and then analyze the resulting esophageal-derived hair follicles. We first confirmed that indeed in the SOX9 loss of function hair follicle, there was a reduction in SOX9 expression. But more interestingly, we found that the SOX9 loss of function by itself was enough to enhance lineage conversion, as you can see by the upregulation of CD34. So altogether, this data suggests that HIF1 alpha and SOX9 may be playing together, may be acting together to prevent lineage conversion. So overall, we've shown that when esophageal cells are exposed to the skin microenvironment, a proportion of them undergo lineage conversion, while regenerative signals, and specifically the HIF1 alpha SOX9 axis, keep cells away from regeneration, promoting a regenerative state. However, it seems that by lifting this barrier, cells can then exit the regenerative state and progress towards this conversion into skin identity. And with that, I would like to acknowledge everyone in Maria Alcolea's lab, especially Maria for her supervision and support throughout my PhD, Maite Bejar, who has been heavily involved in this project, all our collaborators, and the Wellcome for funding.